Hello, this is our third video in the development unit. And in our first two videos, we talked about some fundamental processes that underlie development, including cell differentiation, which is caused by different expression of genes in different cells. And so we talked about the mechanisms behind that. We also talked about some ways that early embryos become different from each other, such as having different cytoplasmic determinants. And we talked about induction in embryos. And then we also used the fruit flies as an example of pattern formation. So in this video, we're going to get more into animal development. And we're going to be in chapter 47 for this. And so these are our learning objectives for today. I want you to be able to summarize the initial stages of development common to a lot of different animal species. So there's kind of four main stages that we'll look at. And we're going to focus today on fertilization. So I want you to be able to describe or illustrate the steps that occur during fertilization in sea urchins and then compare that to what happens in humans. So that's where you get the diverse animal species. Okay, so first of all, let's talk about how different animals can often be very similar in their early embryos. And so here's a picture from the beginning of your chapter, chapter 47, and it shows a chick embryo and a human embryo. And you can see that there are some striking similarities. You can see that there is this early development of the structure that will become the backbone, and you can see some of the organs developing. You can see the eye developing. Now, these are at different stage or different ages. So the human embryo is 41 days old when it gets to this stage, and the chick embryo is only 3.5 days old. But we call this the phylotypic stage, which is when these embryos show their greatest similarity. And also, when we talk about a lot of different animals, a lot of animals are going to have these same four initial stages of embryonic development. And here we're going to show not a chick or a human, but we're going to show a frog. And so frog egg fertilization is the first stage. And fertilization is a fusion of an egg and a sperm, and that's the process we're going to focus on today. After that process, there is a bunch of rapid cell division. So when this zygote begins to divide, that's the end of fertilization and the beginning of the stage that we call cleavage. And cleavage just means to divide. And so we'll have this initial zygote that's formed. It will divide first in half, then each one of those in half, and eventually you'll get a ball of cells that's hollow on the inside, and that is called a blastula. The next stage is gastrulation, which is where this ball of cells rearranges itself, and cells are going to move in this stage. Some of the cells are going to move toward the inside. And so that rearranges the embryo and produces the embryonic germ cell layers. And at the end of that, we have what's known as a gastrula, which has the 3 germ cell layers. And then the next stage is organogenesis, which is when the organs begin to develop. So far we've talked about 3 different organisms that we study embryonic development in. And I just want to talk about the idea of model organisms. Model organisms have been really important in the study of embryology as well as lots of other things in biology. A model organism refers to a species that is chosen to be studied because it's easy to use this species. Many times these species divide quickly, reproduce quickly, and grow quickly, and so you can grow many generations in a short period of time. Many times they are small, and so you can house them in small areas in the lab. Um, so these are some of the model organisms that are used in developmental biology research and in other biology research. We talked about the fruit flies in our second video. We talked about pattern formation, the development of the anterior posterior axis in the fruit flies. The genus and species name is Drosophila melanogaster, and you can see from this picture that you can buy this kit from Carolina Biological Supply Company, and it has everything that you need for growing and raising Drosophila. So these little tubes are what you can house the fruit flies in. And I used to do this all the time with my AP biology students. And and so they don't take very much space, and you can um, easily combine them and mate them, and they can produce offspring. And this all happens within weeks to months. Another common organism that is studied in development is Caenorhabditis elegans, or C. elegans, which is a nematode or roundworm. And we'll talk about that later. We'll talk about sea urchins today. Sea urchins are in the phylum Echinodermata, which literally means spiny skin. And we've learned a lot about the process of fertilization by studying echinoderm fertilization. And then our previous picture on the previous slide was of frogs. Particularly the genus Xenopus is studied in developmental biology research. Xenopus is a model organism for a lot of developmental studies. And in fact, Dr. Brian Hyatt, who teaches at Bethel, he studies development in Xenopus, and he's particularly interested in the development of the lungs. And it turns out that the genes that are involved in early lung development in the frogs are the same genes that are involved in lung development at the early stages in humans. So the frogs are a great model organism. And all of these that I've mentioned here, these first four, all of these have external, well, I guess the fruit fly has internal fertilization, but the eggs are laid outside of the body in all of these, and we can easily observe the development because it happens outside of the body as opposed to in mammals, where development is happening inside the female's womb. Another commonly studied organism in development are chicks, the chick embryos. And again, you can carefully remove the eggshell and you can see what's happening with the chick embryos because they kind of sit on top of the yolk in the egg. So moving then into fertilization. And again, we'll study sea urchins. Here's a picture of a sea urchin, one type of sea urchin that I found. This came from the Moat Aquarium website. And so sea urchin eggs are laid in um, in shallow seawater, and the eggs are covered with a jelly coat. There's no eggshell, but there is a jelly-like coat around them that protects them, keeps them from drying out if the tide goes out, for example, protects them from abrasion from the sand. And the jelly coat secretes chemicals that attract sperms. And so then the sperms will swim toward the eggs. Now, I want to talk a little bit about the structure of a sperm. This is a picture of a human sperm cell, which I pulled from chapter 46. But the sperms of the echinoderms are going to be similar in that they're going to have a head. They're going to have this mid piece that has a lot of mitochondria. They're going to have the tail. And the whole thing is surrounded by a plasma membrane. This is, remember, just one cell. So the head contains the nucleus. The nucleus contains the DNA. That's really important. That's the main contribution of the sperm to the next generation. The head also, in the tip of the head, the anterior end of the head, that part of the sperm cell is called the acrosome. And we're going to see that there are vesicles in this acrosome that hold some digestive enzymes, and these are going to be important in the process of fertilization. So when the sperm it's been attracted to the egg by the chemicals that have been released. And by the way, this is yet another example of cell-to-cell communication, this release of the chemicals. The sperm head then is going to come in contact with the surface of the egg, and the egg, remember, is covered with this jelly coat. In fact, I'm going to show you the next slide and see, you can see that here's the egg. The egg is much larger than the sperm. This is just a piece of a sperm. This is most of a sperm. This figure from your book shows a sequence of events that happens. And so let's just look at the structure of the egg. This is the egg. This is the egg cytoplasm. The surrounding the egg is this jelly coat. And the outside of the egg is going to be the plasma membrane that surrounds the egg. In between the jelly coat and the egg plasma membrane is this purple layer, at least in this diagram, it's purple. And this is called the vitelline layer that you can see labeled right here. Embedded in the egg plasma membrane are some receptor proteins. And so we're going to have a cell-to-cell communication that happens in the process of fertilization. But I'll go back to the words. So when the sperm head contacts the egg surface, this initiates what is known as the acrosomal reaction. What happens in the acrosomal reaction is the acrosome is going to discharge the hydrolytic enzymes that are contained in the acrosome, and these enzymes will begin to digest the jelly coat and make kind of a hole in the jelly coat. Also, once the hydrolytic enzymes are released, then the acrosomal process can form. And we'll go back to this picture again. There are some actin filaments, some actin protein filaments that are here in between the nucleus and the acrosome of the head of this sperm. And once the acrosome enzymes are released by exocytosis, then these actin filaments, microfilaments, can begin to grow, and they push the plasma membrane of the sperm cell out. And so you get this process, this extension of the sperm. And so that is called the acrosomal process, an extension of the sperm cell. It elongates and it penetrates the jelly coat. Then there is a cell-to-cell communication, a direct cell-to-cell communication between the acrosomal process and the plasma membrane of the egg. So there are proteins on the surface of the acrosome, and these proteins can bind to the receptor proteins that are embedded in the egg cell membrane. So we have a direct communication happening there. This direct communication causes, well, I'll go on to the next slide and show you in words what this causes. The recognition between the sperm acrosome and the egg, this binding between the proteins on the sperm acrosome and the receptor proteins on the plasma membrane of the egg, this triggers fusion of the two plasma membranes, the fusion of the plasma membrane of the egg and the sperm. So we can see that here. We can see that this plasma membrane of the egg and the plasma membrane of the sperm have now fused. And this allows the sperm nucleus to enter into the egg cytoplasm. Also, what is triggered is some opening of some ion channels in the egg plasma membrane. This allows ions to rush into the inside of the egg, and this depolarizes the membrane, very similar to what might happen in a neuron. So we have an electrical depolarization. This depolarization makes it impossible for other sperms to enter the egg. And so this is called a fast block to polyspermy. Polyspermy would be if multiple sperms enter the same egg. This would be a bad thing because remember, each egg is haploid and each sperm is haploid. So each egg, well, I'll talk about a human egg just because I know the chromosome numbers. Each egg has 23 chromosomes. Each sperm has 23 chromosomes. So if you combine an egg and a sperm, then you get 46 chromosomes, which is the normal diploid number. And each of those chromosomes would have a homologous pair that it could line up with in a meiosis. Well, if you had two sperms enter, you would end up with 23 plus 23 plus 23 is more than 46. It would be 69 chromosomes. And that would cause big problems in the development of the embryo. And in humans, if you have an extra sperm, then that embryo is not going to develop at all. Probably the same case with the sea urchins. So they block polyspermy with this depolarization. Then the next thing that happens are changes within the outside edge of the egg cell. So the outside of any organ or any cell is called the cortex. You've probably heard of your cerebral cortex. Well, that's just the outer layer of your cerebrum. Your kidneys also have a cortex. So a cortex is just an anatomical word that means outer layer. So there are some vesicles in the outer layer of the egg, and we will go back again to this slide, and you can see these vesicles in this cortical area, the outer area or the cortex of the egg. And so these vesicles are called cortical granules. These vesicles are going to be stimulated to fuse with the plasma membrane and release their contents outside of the cell by exocytosis. Some of the contents of the cortical granules are enzymes, and these enzymes are going to digest through the sperm binding receptors, and they're just going to cut off these sperm binding receptors. This allows the vitelline layer that we talked about to be lifted away from the plasma membrane, and this forms a fertilization envelope. So again, if we go back to the picture, you can see these receptor proteins, and they are kind of holding the vitelline layer on to the plasma membrane. And so if you snip those off, then they're no longer going to be able to hold this vitelline layer. And also some fluid is going to seep in here, and that is going to push the vitelline layer away from the plasma membrane. And the vitelline layer then becomes hardened and becomes what we call a fertilization envelope. Now, it is thought, or some researchers hypothesized, that calcium ions might play a role in the fusion of these cortical granules to the plasma membrane, because we have already seen that calcium signaling controls fusion of vesicles with the membrane in neuron signaling. So when action potential arrives at the end of an axon, some calcium channels are open, calcium floods into the cell, and this causes the vesicles in the axon to fuse with the membrane and release their contents into the synapse. So researchers hypothesize that something similar might be happening with a sea urchin egg. And so here are some pictures that they took of sea urchin eggs at different times after adding some sperms to some eggs. You can see some sperms around the eggs. So 10 seconds after fertilization, none of the sperms have been able to digest through that jelly layer. But One of them apparently got through up here, and so we see a fertilization envelope starting to form at about 25 seconds, and that fertilization envelope spreads. And so by 35 seconds, it's spread a long ways. And by a minute, that fertilization envelope has surrounded the entire cytoplasm of the cell. And so there's a space between the egg cell membrane and the vitelline layer. And so this is a fully formed fertilization envelope. These lower pictures down here are showing the calcium concentration. So 1 second before fertilization, and so these times don't line up perfectly with the times that are shown up here. But 1 second before fertilization, this is going to be the concentration of calcium ions. There's some calcium, but there's not a ton of calcium. As soon as the sperm enters, 10 seconds after its entry, a bunch of calcium ions have flooded into this cell. And now we have a high concentration of calcium here. And that kind of correlates to the beginning of the fertilization envelope there. And then the calcium ions just spread through the whole cell as the fertilization envelope is spreading. And eventually the calcium ions have gone all through the cell, and the fertilization envelope can surround the whole cell. So this was really good evidence that calcium signaling is involved with the fusion of these cortical granules. Okay, once the egg is once the sperm enters the egg, once the sperm nucleus enters into the egg cytoplasm, this initiates certain chemical reactions within the cell, and it also speeds up certain chemical reactions. These reactions include cellular respiration, and cellular respiration is what happens inside the mitochondria to produce lots of ATP. And we've learned some of the uses of ATP when we talked about cell signaling, but ATP is used for all kinds of cellular work. So in order for the cell to begin to divide and grow, we need a lot of cellular respiration to produce lots of mitochondria. We also need lots of protein synthesis. We need our ribosomes to start making lots of proteins that are involved in cell division. And so this ramping up of these metabolic reactions is called activation. When the egg is activated, then it's ready to begin dividing. And it's not until after activation that the egg and the sperm nucleus fuse. In fact, it takes about 20 minutes in the sea urchin between when the sperm enters the cytoplasm and when the egg and the sperm actually come together. And once the egg and sperm nucleus fuse, then they can start having cell divisions. It's been found that activation can be triggered by an injection of calcium, even when there's no egg nucleus present. So you can just inject some calcium ions, and that will trigger the increase of these metabolic reactions inside the cell, and it'll get the cell ready to divide, even though there's no way it's going to divide because there's no egg nucleus. And the fact that you can trigger activation by the injection of calcium suggests that the proteins and the messenger RNAs are already there in the cytoplasm. You don't have to have transcription in the nucleus and then translation. There's going to already be a lot of mRNAs and already a lot of translated proteins in the cytoplasm. Okay, so this is what happens in the sea urchin, and I'll just go back to this picture again. This is what happens in the sea urchin. We have the acrosomal reaction, so the release of the acrosomal enzymes and the formation of the acrosomal process, and then the fusion of the egg and the sperm cytoplasmic or plasma membranes, And then the cortical reaction, which is the fusion of these cortical granules and release of their enzymes and the clipping off of these receptors, and then the formation of the fertilization envelope. So this is what happens in a sea urchin. And what we want to look at now is how this compares to what happens in humans. So one thing that's different in humans, in all mammals, in fact, is that fertilization is going to happen inside the body. In humans, you know that it happens in the fallopian tube. But before fertilization can happen, the egg has to be ovulated from one of from an ovule in the ovary. And after ovulation, an egg contains some, and I'll go to our next slide to show you this. Um, an egg is going to be surrounded by some of the follicle cells from the ovary, which are going to adhere to the egg. Another difference is that there's not a jelly coat, and instead the extracellular matrix is called the zona pellucida. So there's still kind of a layer that the sperm has to penetrate through in order to get to the egg plasma membrane. But the layer is different. Instead of a jelly coat, it's going to be these follicle cells and then the zona pellucida. So we'll go back. So the sperm has to travel through the follicle cells and through the zona pellucida. And an acrosomal reaction is going to facilitate this. So the acrosome, just like in the sea urchin sperm, the mammal or human sperm is going to release acrosomal enzymes, and they are going to help to digest a pathway through the follicle cells and through the zona pellucida so that the sperm can contact the plasma membrane. And once the sperm contacts the plasma membrane of the egg, then the egg and the sperm nucleus can fuse, and this is going to cause the release of calcium ions again, and this is going to cause a cortical reaction, which is going to cause a slow block. In mammals, there's no known fast block, so they don't have the depolarization that causes the fast block, but they do have the same cortical reaction that causes the slow block. So the zona pellucida will be lifted away from the egg cell membrane, the egg plasma membrane, and so there'll be a space there, and so that'll prevent any more sperms from getting in. One thing that is different in the mammals versus the sea urchins is that fertilization is much slower in mammals. Depending on the species, the first cell division will happen 12 to 36 hours after the initial sperm binding with the plasma membrane. But in sea urchins, it only takes 1.5 hours before the sperm binds. I'm not really sure why this is true, but that's another difference between fertilization in mammals and fertilization in sea urchins. Okay, so just one more look at the full-size picture here. Again, we're going to have an acrosomal reaction that helps the sperm get through the zona pellucida. And I tried to read up a little bit on this, and it seems like that acrosomal reaction either starts here in amongst the follicle cells, or it starts once the sperm contacts the zona pellucida. But at any rate, the sperm is able to digest its way to the plasma membrane. And you see here that these membranes have already fused, and the sperm nucleus is about to be released into the cytoplasm of the egg. And you can also see that the cortical granules are releasing their contents, and this is going to cause the zona pellucida layer to pull up away from the the egg cell membrane. Okay, so that is that covers fertilization, and this is where we will stop today. And in our next video, we will look at cleavage, which is the next stage in embryonic development.